Okay, class, one of the first things we're going to talk about is the concept of experimental design. Um, you guys probably in grade school, middle school talked about the scientific method. So it's the same concept. Experimental design and scientific method are, in my mind anyway, one and the same. So we're going to talk about the different approaches that scientists take when they're looking at a different problem, or when they have a certain purpose, something we want to look into. Um, so we're going to approach these different problems in this class anyway, using experimental design, scientific method. One of the examples that I gave to you, just so that you can kind of see the methodology that we're going to use. Uh, if you were to stumble across a bunch of bats on a cave floor that were dead, or find some scattered in a certain location that were dead. And you also noticed something about them. They have a white muzzle, a whitish color on their muzzle. You might make that observation, and that's many of the scientific things begin with an observation. So that would be an example of an observation. White muzzle, lots of dead ones. So you're going to look into that, and you're going to maybe make a hypothesis. A hypothesis is something that includes if, then, and because, right? So kind of a loaded hypothesis in this case would be if we can determine the cause of the white color on the bat's muzzle, we can determine the cause of the bat population's high mortality, the reason they're dying. Because we only see high levels of mortality in cave systems where we see this white color on bat population's muzzles. So where we're seeing a lot of white muzzles, we're seeing a lot of dead bats. We think there's a relationship there. That would be a hypothesis. So something that you could start to look into. And at this point, you might want to do some initial research, look into some things. So is this something that we see in south-facing caves, or north-facing caves, or deep caves, or shallow caves, or other things? There's other things that you could look into: warm caves, cold caves. There's lots of different things that you could look into. You guys think for a minute, what would be some of the different things that you could look into before you actually start your research? We're going to design an experiment, but in order to design an experiment, we kind of have to cut out everything that's going to cloud the results. So we have to have certain controls. And what do you think would be some necessary things that we have to do if we're going to design an experiment that's going to determine the cause of bats dying? What are some things that we would want to make sure that we keep the same? Scientists uh, have to approach problems very carefully so they're not clouding their results. All right, so we want certain controls. One thing we might take into consideration is that there are a lot of different species or kinds of bat. So are we seeing the white muzzle on lots of different species, or are we only seeing them on certain species? And are we going to study every species, or are we going to limit only to certain species? What about caves? Are we gonna study caves, bats that come from, say, Northern California or the Cascade Mountains? Or are we gonna study Oklahoma? We're gonna study warm caves. We're gonna study cold caves. So we have to make some perimeters before we do an experiment. Then we have to decide what our dependent variable is. Okay, so it's going to depend on something, right? Dependent variable. The number of deceased bats that field researchers collect depend on what? So is it depend on the frequency of this white muzzle that we see? And there's an independent variable. What type of data would a field researcher collect? So your independent variable and your dependent variable, and they both play a role. Your independent variable sometimes is something as simple as time. It depends on how long you've been doing it. That's going to be your independent variable. So scientists, we're going to stray away from bats for just a second. Scientists use a lot of different techniques when they're presenting data. You see line graph there, illustration, a map. they're going to analyze data, collect data, and then present data in different ways. And in the way they present their data, you should be able to see their independent variable and their dependent variable. Here's a couple of different ways scientists present data. There's a pie chart right there that shows world population percentages by country. So as you can see, the big chunks down here, China and India. In the middle here is a data table for frogs, measuring the circumference of a thigh on a frog leg. Somebody felt compelled to study that. You've got more applicable and recent data here, COVID-19 right up there in the case. So that's what's called a scatter plot. And you can see multiple things all on one graph. So these are two different types of graphs. And then down in the lower left there, you have a bar graph. So just a side-by-side comparison of different things. Okay. You've got your independent variable on the x-axis. That's the axis that's going horizontal. And then your dependent variable on the y-axis, the up and down axis. And you can see which thing's an independent variable and a dependent variable. So in this scatter plot in the upper left-hand corner, you see eruptions of Old Faithful, which is a geyser in Yellowstone Park, and then the waiting time between. That's going to be your dependent variable. You see a data table down below that just shows different incidents when they're looking for oil and fracking. You see another graph over here that shows temperature, and it looks like ice cream sales. So you can see that ice cream sales go up based on the temperature. The independent variable here would be the temperature. So the hotter that it gets, well, the sales depend on the temperature. Down below here, the independent variable is going to be the date, and then it shows the number of cases of COVID-19. And it goes up and peaks the longer it's been in that area. Yeah. This is a bat graph. So if you look at a few different ones, let's take a second here. Let's look at this one. What is the independent variable and what is the dependent variable? Take a second to think before I tell you what it is. See if you can beat me to the punch. What's going to be your independent variable and your dependent variable? You've got the cumulative probability of extinction. So the likelihood that this actually applies to bats, and then years into the future. So 20 years from now, 40 years from now, 60 years from now, what's the likelihood that an animal is going to be extinct? So what's your independent variable and your dependent variable? Okay. So the probability of extinction is going to go up in years into the future. So your independent variable is going to be the years into the future, and the probability of extinction is going to be the dependent variable. Okay. What's going to be the independent variable and the dependent variable here? There's two different graphs here. They show different ethnicities and the frequency of COVID-19 since it's come out in March, April, May, June, and August. And you can see in some different groups, the frequency has gone up, and in other different groups, it's gone down. So what's going to be your independent variable and your dependent variable there? Yeah. Last one we'll talk about here. What's going to be your independent variable and your dependent variable here? This is active cases per day, 14-day. Okay, so the purple is going to show cases of COVID-19 in a particular county overall. They're saying that if you have COVID-19, it takes you 14 days to get over it. So they register you for 14 days. And then the little blue down at the below shows new cases as they pop up. So if you just look at this data, what's going to be your independent variable and your dependent variable? Okay. The number of cases is going to be your dependent variable. And it's going to depend on when you collect your data, right? So the month is going to be your independent variable. So dependent variable is going to be on the y-axis. Independent variable is going to be on the x-axis. Okay. So let's say we've done our experiment, we've presented our data. Once the data has been analyzed, let's go back to the bat problem here. Let's talk about bats again. Scientists decide whether or not they want to proceed forward with a theory, or do they want to reevaluate, design a new experiment. So sometimes they'll do an experiment and it won't exactly result in a theory. There's still some cloudiness. So if scientists have determined that there's a connection between the white muzzle on bats and the incidence of mortality, then they can begin to research the cause of the white muzzle. It'd be totally unnecessary to study the little white patches on the muzzle of a bat if there was no link between mortality and the white muzzle. So scientists now have the ability to pose further research questions. Now, sometimes you do an experiment and then you have a whole bunch of other questions that you have to research, and that's just science. That's science. That's what makes science kind of interesting. Sometimes you turn over a rock and then you go, whoa, I've got 100 more rocks I need to turn over. So if you look here, here's just some questions that could be derived from, you know, doing this experiment that we just did with the bat muzzle and the white muzzles on bats. What causes the white muzzle on certain bat populations? So why do some species of bats have it and some don't? Why do some species get it, some don't? Another thing is the white muzzle caused by diet deficiencies, or is it a fungus, a virus, a bacteria? Does the white fungus only occur in certain locations, temperatures, and climates? Or is it everywhere? Just everywhere in North America? When does the white muzzle show up in a bat population? Is this something that pops up in the summertime, or is this when they go into hibernation in the winter? Can the cause of the white muzzle be addressed or cured or prevented? Can you do anything about it? What's the future for affected bat species if it can't be cured? Yeah. And then how would losing these bats impact inhabited ecosystem? So if all these bats went extinct, what would happen? I know just here around here, if we didn't have bats, we'd have a lot more mosquitoes. It's kind of nice living in a place with not very many mosquitoes. At least I think it is. So in real life, they have done a lot of research on this, and that bat, the white muzzle, is caused by a lethal fungus, and it does only affect certain bats. It's bats that hibernate in cold caves, like the ones that hibernate in the Cascade Mountains. Oregon's bat population is very threatened by a fungus that causes that little white muzzle. So scientists are never done, and the research builds on the research, which builds on the research. So once they uncover maybe a cause, then their next question is they have to figure out, what to do about it. And how would you approach it? And so they'll gather up bats that are living, and they might even put them in lab situations, and might even give some a fungus and try to figure out different ways to cure it. So might be sacrificial bats in the next experiment. Anyway, the research is never done, and this is how we get things like vaccines and inventions and progress. This right here is the scientific method. Okay? Control, independent variable, dependent variable, experimental design. So we're going to see it a lot this year.